Cells are the building blocks of life, but not all cells are the same. Today, we'll explore two major cell types, prokaryotic and eukaryotic, covering differences from basic to advanced levels. Let's start with the basics. Prokaryotic cells do not have a true nucleus. Their genetic material floats freely in the cytoplasm, in a region called the nucleoid. Eukaryotic cells have a well-defined nucleus enclosed by a nuclear membrane, which protects and organizes the DNA. Prokaryotic cells are typically small, ranging from 0.1 to 5 micrometers. Eukaryotic cells are larger, generally between 10 to 100 micrometers. Prokaryotes have a simple structure, lacking membrane-bound organelles. They mainly contain ribosomes for protein synthesis. Eukaryotes contain membrane-bound organelles like mitochondria, Golgi apparatus, and the endoplasmic reticulum, making them more complex. Structural differences. Most prokaryotic cells, such as bacteria, have a rigid cell wall made of peptidoglycan in bacteria or other materials in archaea. In eukaryotes, a cell wall is present only in plants composed of cellulose and fungi composed of chitin, while animal cells do not have a cell wall. Both prokaryotes and eukaryotes have a plasma membrane composed of a phospholipid bilayer, but in eukaryotes, the membrane may contain more complex lipid structures like cholesterol. Prokaryotic ribosomes are smaller 70s compared to the larger eukaryotic ribosomes 80s. Despite their size difference, both ribosomes perform the essential function of synthesizing proteins. Prokaryotes have circular DNA called plasmid, which is not wrapped around proteins without histones except in some archaea. Eukaryotic DNA is linear and organized into chromosomes within the nucleus, tightly wrapped around histone proteins to form chromatin. Prokaryotic cells divide by binary fission, a simple process where the DNA replicates, and the cell splits in two. Eukaryotic cells undergo more complex division processes, mitosis for growth and repair and meiosis for sexual reproduction, involving the organization of chromosomes and several phases. In prokaryotes, energy production occurs in the cell membrane since they lack specialized organelles. For example, the electron transport chain for ATP synthesis occurs along the plasma membrane. Eukaryotes use specialized organelles such as mitochondria for aerobic respiration and chloroplasts for photosynthesis in plants for energy production. In prokaryotes, transcription and translation occur simultaneously in the cytoplasm. As soon as mRNA is transcribed, ribosomes start translating it into protein. In eukaryotes, transcription occurs inside the nucleus, and the mRNA is processed, spliced, capped, and tailed, before it is transported out of the nucleus for translation in the cytoplasm or on the rough earth. Prokaryotes often regulate genes in clusters called operons, e.g., the lac operon in bacteria, where multiple genes are transcribed together under a single promoter. Eukaryotic genes are more complex, typically controlled individually with separate promoters, enhancers, and a variety of regulatory proteins. Both prokaryotic and eukaryotic cells are essential for life on Earth. Their differences not only highlight their unique characteristics but also their evolutionary journeys. Understanding these cells gives us insight into the vast diversity of life. If you found this video helpful, don't forget to like, share, and subscribe to our channel for more engaging science content.